The subject that I'm going to discuss during this presentation is the effect of infection, especially viral infections during pregnancy, with a focus on the role of the placenta in the response to the infection as well as into the protection of the mother and the fetus. Before I start, I want to acknowledge that much of the data that I'm going to show during this presentation was produced by Palomi Aldo, Drs. Jae-Yong Kwan, Karen Rasicot, Ingrid Cardenas, Yuhi Ding, Michelle Silasi, and Yuan Yu. So what do we know about infection and pregnancy? There is a lot of data, especially epidemiological data, showing that infection represents an important and frequent reasons or causes of pregnancy complications. And the classical example is preterm birth. 40% of preterm births occur to mothers with intrauterine infections. And we know that preterm birth, as an example, is a major burden, not only because it affects the health of the mother, but also the health of the child. And in addition, is a major burden to the healthcare system. So, in general, the problem of infections and pregnancies, which are now elucidating every time understanding better and better, is extremely important for the protection of the mother and the fetus. And indeed, when I talk about the protection of the mother and the fetus, we know that infections during pregnancy may have a major effect on the mother, including the induction of maternal mortality. And there is now growing evidence that infection during pregnancy will have also an effect on the fetal development. So let's start discussing very quickly about these two types of effects. Let's start with infection and pregnancy and the maternal effect. We know for many years that pregnant women are more likely to experience severe complications, mortality associated with viral infections. And the cases is, for example, during the pandemics. During 1957-58 influenza pandemic, 50% of the deaths occur to pregnant women. And more recently, pandemics like H1N1 or Ebola, the pregnant woman is a high-risk population who is affected in a higher percentage than the rest of the populations. And as I mentioned, the most recent H1N1 pandemic, there was a significant number of pregnant women who died during the viral infection. Interestingly, the deaths of those cases were always associated with secondary bacterial complications. So indeed, pregnant women represents now a high-risk population during viral infections. In the fetal side, viral infections during pregnancy has been shown to correlate with increasing fetal development abnormalities, including brain development. And there are several studies showing that during times of pandemics, there is an increase in the number of children with autism, with schizophrenia, and more recently, what I think is a clear evidence that virals may be detrimental to pregnancy is the cases of microcephaly due to Zika. The information associated with these emergencies, with these pandemics, is not something of the past. It's something that we will be confronting every time more and more as the exposure to different viruses in populations that will never had interaction with viruses is just increasing. So one of the major questions that has been brought looking at those epidemiological studies is why pregnant women are more susceptible to infections. And an early answer to this question was an old dogma that has been present for many years. That is the fact that the maternal immune system during pregnancy is suppressed. And therefore, the increased mortality during the pregnancy is due to this maternal immune suppression. The fact is indeed that during pregnancy, the maternal immune system invades the uterus, and you can have at the deciduum, and as well as in the whole uterus, almost all the different types of immune cells that we know about, including macrophages, NK cells, T helper cells, dendritic cells, and so on. And the presence of the immune cells at the implantation site was considered as a proof that there is a response of the maternal immune system against the fetus, because the fetus is considered a semi-allograft with paternal antigens that are able to trigger a maternal immune response. This was proposed and recognized by Sir Medawar more than 50 years ago, that proposed this paradox that the fetus as a semi-allograft needs to escape the rejection by the maternal immune system. And the way that the fetus escape this recognition of the paternal antigen is by inducing an immune suppression during pregnancy. But the side effect of this immune suppression is that the woman is more susceptible to infections. The reality is that in the last 10 or 15 years, we have recognized that this hypothesis is incorrect, because the maternal immune system during pregnancy is not suppressed. It's the opposite. It's stronger than ever. And why that? Because a systemic immune suppression during pregnancy represents a danger to the species. A pregnant woman who will be, if all the pregnant women will be immune suppressed, they will be more susceptible to any environmental stress, and the species will be eliminated. And we know in addition that the high number of immune cells that are recruited at the implantation site, they are not there because they are attacking the pregnancy. They are there because they are necessary for the success of the pregnancy. The immune cells recruited at the implantation site, they are essential for the different stages of the pregnancy, including implantation, trophoblast invasion, as well as parturations. And it has been shown that depletion of immune cells at the maternal-fetal interface, instead of helping the pregnancy, will have a major detrimental effect. And just to give some examples, if we deplete natural killer cells, there will be problem with the transformation of blood vessels. If there is depletion of macrophages, there will not be trophoblast invasion, as well as if there is no Tregs, of course, there is no tolerance, and if there is no dendritic cells, there will not be implantation. So, to put this whole picture together, the maternal immune system is not suppressed during pregnancy. It requires a functional maternal immune system for the success of the pregnancy. And in addition, as we discuss in a few minutes, the fetal placental unit is exposed to microbial products coming from the mother. So it needs to have an active and a strong immune system to protect the fetus. So that has led to the re-evaluation of the type of immunological response that we have in pregnancy. Is really pregnancy a graft host immune response, or is maybe an immunological response associated with what we see in the tumor host immune responses? And more data suggest that indeed pregnancy is not a graft host disease, but it's more like a tumor immunology. And it's interesting because many processes that we can see in tumor development are very similar to what we see in pregnancy. And just to give an example, during the process of implantation, implantation requires an inflammatory response that allows the trophoblast to attach, invade, induce the production of blood vessels, and attract the immune cells. Exactly the same process, exactly the same inflammatory process we can see during tumor during the metastatic process. But more important, I think, is the aspect of the induction of tolerance or antigen presentation. If we compare the host graft disease and the tumor antigen presentation process, we can see that both are completely different. So let me go quickly through the two of them. During the graft has this model, we are taking an organ which already developed, who has high number of antigens, but more important, it has its own dendritic cells or antigen presenting cells. When this organ is transplanted into host, the transplanted dendritic cells will be carrying, as well as the host dendritic cells, will be carrying a high number of antigens. In other words, a high load of the antigens from the graft. Those antigen-presented cells will migrate into the lymph nodes, where will present this high amount of antigens, and of course will induce a classical immunological response related to the rejection. The tumor, on the other hand, starts always with a small number of cells. And that small number of cells represents a small amount of antigens. That's the first aspect. The second aspect is that the host antigen presenting cells, in this case the dendritic cells, will take that small amount of antigen, will carry to the lymph nodes, and because of the load of the antigen and the lack of additional signals that I don't have time to discuss, the outcome of this is the induction of tolerance to those antigens. And the final, of course, is induction of tolerance to the growing tumors. If we look at the pregnancy never happens as a transplant of a big organ carrying its own antigen-presenting cells. In reality, pregnancy starts with one cell or two cells or very few cells. At the implantation site, those few cells start releasing a small amount of antigens. That small quantity of antigens is taken by the host, in this case the mother antigen-presenting cells or dendritic cells, who will carry those antigens into the lymph nodes. And at the lymph nodes, similar to what happens with the tumor, because of the load and the type of presentation, will induce a tolerance to those antigens. So clearly, what we see is antigen presentation during pregnancy resembles a tumor antigen presentation rather than a graft antigen presentation. Let's talk quickly about the immune modulation. So the tumors are known to be loaded what is called the tumor immune infiltrate. Why that tumor immune infiltrate is not rejecting the tumor? And the reason is because the cancer cells are secreting immune regulatory factors that will induce the differentiation of those immune cells or will control and prevent any anti-tumor response. Very similar is happening during pregnancy, where we have the trophoblast, the cells from the placenta, secreting immune regulatory factors. And those factors at the implantation site will induce this unique differentiation of the immune cells. And those cells now will protect and help the pregnancy. So definitely pregnancy is a unique immunological condition that is associated with a specific antigen presentation as well as immune regulation. The second aspect that also makes unique the pregnancy is the interaction with bacteria. Bacteria present at the uterus, especially in the lumen and the lower reproductive tract, has always been associated with a risk for the pregnancy. And indeed, we know that bacteria can ascend from the lower reproductive tract into the uterine cavity, reach the membranes, the placenta, and even invade inside of the fetal cavity of the membranes. And when does it happen, it will induce fetal death. And there are pregnancy complications associated with bacterial infections, such as chorioamnionitis, decidulitis, and so on. And there is no doubt that all of them are due to bacterial infections. However, we know that the presence of bacteria is not enough to induce inflammation, fetal death, or pregnancy complications like preterm birth. Use of antibiotics in order to prevent pregnancy complications has not been always successful. And moreover, we know that there is intrauterine infections that are identified even in the absence of bacteria in the amniotic fluid. There is growing evidence that there is a normal microbiome that is present in the reproductive tract and that the uterus is not a sterile environment. We did experiments several years ago where we took Escherichia coli, E. coli, that was introduced a green fluorescent gene so we could be able to track that bacteria wherever we put it. And the place where to put the E. coli GFP is in the vagina of the mouse, as I'm showing here. After the delivery of the E. coli in the vagina of the mouse, we follow the movement of this bacteria within the uterus of the mouse. And what I'm showing here is, of course, it's showing the background that we don't see GFP in the uterus. After delivery of the bacteria of the E. coli, the GFP E. coli, we see around 12 hours that is concentrated in the vagina, which it makes sense because that is the place where we deliver the bacteria. And now look what happened after 24 hours. The bacteria has moved through the whole uterus of the mouse and even has reached the ovaries. 48 hours, the signal is very intense, again, in the two horns of the uterus of the mouse as well as in the ovaries. 72 hours, that bacteria is now controlled. The signal has been reduced in the uterus, leaving only a strong signal in the vagina. However, when we look by PCR, if there is still bacteria left in the uterus of the mouse, the answer is that yes. Looking at the presence of GFP, we see that in 12 hours, 24, 48, even in 1 week, after when the signal, or the GFP signal by fluorescence is not detectable, by PCR it is detectable, suggesting that bacteria is still present in the lumen of the mouse. So in other words, what we are finding is that this uniqueness of the pregnancy compares two components. One is the immune system that is present in the deciduum, and potentially bacterial signals, the commensal bacteria that is sending signals, commensal bacteria present in the lumen of the uterus or in the maternal circulation, that sending signals that are recognized either by the placenta or by the immune cells that are present at the implantation site. So, putting together this immunological and microbial condition that is unique on the pregnancy, which suggests that in order to survive, mammalian pregnancies have established a symbiotic relationship with the maternal microbiome as well as with the maternal immune system. But this still leaves us with the question, why pregnant women are more susceptible to viral infections. And the answer that we have been proposing for several years is that maybe is the placenta. What is uniqueness in the placenta from an immunological point of view? We consider the placenta as an active immune regulatory organ. And similar like other immune cells, the trophoblasts of the placenta express pattern recognition receptors, including toll-like receptors. Those receptors allows immune cells to recognize bacterial, virus, parasites, in other words, recognize whatever is in the microenvironment. And as a result of that recognition, they will respond in order to modulate or trigger an immune response. In order to understand whether the placenta plays an immunological role during infections, the objectives that we defined in our studies were the following. One, to identify or characterize the capacity of the placenta to recognize signals. The second, how the placenta responds to those signals. The third, how the placenta protects against the virals and bacterial infections. And the last one, how the bacteria, how the response to bacteria will have an effect or systemic effect in the mother as well as in the fetus. In this presentation, I am mainly going to focus in the two last aspects, and I'm going to show very recent published data that supports or demonstrates the mechanism or the function of the placenta for the protection and how it may affect systemically to the mother. The recognition and response, I will go very quickly because it's data that we have published many years ago. Recognition. So as I mentioned before, the placenta toll-like receptors are a family of membranal and intracellular receptors who have a specific role to recognize either bacterial, viral, or parasite products. In the placenta, as I mentioned, we found all of these toll-like receptors, including the membranals as well as the intracellular. Once the toll-like receptors will recognize bacterial or viral products, they will activate intracellular pathways, such as the MyDDA and NF-kappa B pathway, that will lead to a specific response, in the majority of the cases, the induction of an inflammatory response. We have published several papers describing the characterization of these toll-like receptors in the placenta. Toll-like receptors have two roles. One, as I mentioned, immune response, because they recognize and respond to bacterial and viruses. But they also have immune modulation because they produce signals that contribute to a proper response of the immune system, in many ways preventing that there will be a damage to the tissues where the infection is taking place. So the next aspect that we have been evaluating is what type of responses are induced in the placenta when we stimulate any of these toll-like receptors. So in order to characterize the response of the toll-like receptors, we use first trimester trophoblast cells, either primary cultures or cell lines, and we expose them to TLR ligands and characterize the cytokine profile. One of the first ones that I'm going to show here, and I will use an example of the uniqueness of the response, is the toll-like receptor 4. Toll-like receptor 4, we show in several studies, and many other people have also shown, is expressed in trophoblast cells. And the main role of toll-like4 is the recognition of LPS, lipopolysaccharide, that is a product of bacteria. So what we did is we took trophoblast cells and we exposed them to different concentrations of LPS. So now let me tell you that in general, if we take a macrophage and we expose to LPS, a macrophage through the TLR4 will respond to LPS by the induction of pro-inflammatory cytokines such as TNF-alpha, interleukin beta, and so on. So we look at those cytokines in the trophoblast, exposing to the same concentrations of LPS that we will put to a macrophage. And what we found is that contrary to the classical pro-inflammatory response that we see in immune cells, in the trophoblast, the inflammatory cytokines are decreased instead of induced. But it's not that the TLR4 pathway is not functional in the trophoblast, because we do see a response, although it's a different type of response. And this response was characterized by a significant increase in chemokines such as IP10 and RANTES. But more interesting is that we saw that LPS in the trophoblast was inducing the expression of type I interferon beta. And you can see in this figure there is a significant change in the messenger RNA in response to LPS in the trophoblast. And if we look at the specificity in this response, when we do a dose, when we expose the trophoblast at increasing concentrations of LPS, we also see a classical dose response in terms of the increase in interferon beta expression. And this is at the messenger RNA, and we find also in a timed response that there is an early and fast increase within 120 minutes in the production of interferon beta messenger RNA. And I want to bring to your attention that after this peak of messenger RNA, then the response starts going down, suggesting there are mechanisms that are controlling or suppressing the increase in interferon beta. But most important, it's very specific for interferon beta, because we don't see the other type of type I interferon, that is interferon alpha, induced following LPS stimulation, TLR4 stimulation with LPS. So we can say that a microbial product sort of like LPS binds to TLR4 and is inducing interferon beta very fast messenger RNA. And this is translated also at the protein level, where we can see that there is increased in the intracellular protein expression of interferon beta following LPS treatment between 24, 48, and 72 hours. But extremely important, there is secretion of interferon beta following LPS stimulation, since when we collected the supernatant of the cells and we look for the presence of secreted interferon beta, we see the accumulation of interferon beta starting at 12, 24, and peaking at 72 hours. So the next question that we had is, what is the intracellular pathway that is inducing interferon beta in response of LPS? So let me remind you again that the trophoblast, if we take monocytes, LPS will bind to TLR4, and TLR4 will induce, together with MyD8, the activation of the NF-kappa B. NF-kappa B goes to the nucleus and induces a pro-inflammatory. That is the classical pathway of TLR4. In the trophoblast, we are not seeing that. So the question was, what are the pathways that are induced in the trophoblast? And what we found is the following. When LPS binds to TLR4 in the trophoblast, recruits TRIF, phosphorylase TBK, as you see in the Western blot in the right side. We are always comparing the non-treatment versus the cells who were exposed to LPS. And again, what we see is increase in the phosphorylation of TBK, increase in the phosphorylation of IRF3, And IRF3 is the major inducer of interferon beta. And that's where we see increasing concentrations of interferon beta. If we block the TBK pathway using BX795, an inhibitor of TBK, we don't see the induction of interferon beta, confirming that this is the pathway that is required for the activation of interferon beta following LPS TLR4 activation. So, unlike most immune cells, TLR response, especially TLR4 response in the trophoblast, do not involve the NF-kappa B pathway, but involve the TRAM, TRIP, TBK, IRF3 interferon beta transcription. This was done in vitro. Is this the same happening in vivo? And of course, we went to the mice, we took pregnant mouse, and we exposed them to low concentration of LPS, or physiological concentrations of LPS. And it's exactly as we saw in vitro, we saw also in vivo. There is an increase in interferon beta expression in the placentas of mice exposed to LPS. And if we do the same experiments with mice who are deficient in TLR4, we will see that even the baseline expression of interferon beta is gone. And also, if we remove TRIP, or we use mice who do not have TRIP, we also do not see the induction of interferon beta in those mice. Again, demonstrating that the pathway necessary for the induction of interferon beta in the placenta is TLR4 and TRIP. So we put it together, we can say that bacterial products through the TLRs, especially TLR4, and we know also TLR2, may function as the signals necessary to maintain interferon beta production in the placenta. And this interferon beta is critical in order to maintain the protection against viral infections, but also in order to promote tolerance and prevent excessive inflammatory responses. If any of the components of those signals are removed, the expression of interferon beta can be affected. But then the question is, why interferon beta? Why the placenta is continuously expressing these basal levels of interferon beta? So what do we know about interferon beta? And we know a lot because interferons has been known for many years. Early production of interferon alpha and beta is an important activation signal for immune cells. They produce a lot of regulatory factors that suppress immune responses by enhancing IL-10 and PD-L1 expression. They function as antiviral signals, but the production of these antiviral signals also needs to be maintained in equilibrium, and that is again the other functions of the interferons. So again, in one hand, type I interferons are critical for the antiviral responses, but also are necessary to maintain or they have this immune regulatory, immune modulatory effect. And the balance of these two signals is really what maintains the health and prevents the damage of the tissues. So the next question that we have is, what is the role of the placental interferon? What are the signals that are induced by interferon? So let me review very quickly how interferon beta works. Interferon beta will bind to its receptor, the interferon type I receptors, and it will induce the induction of multiple genes that are known as interferon-stimulated genes. And they have multiple functions. They interfere with stages of viral replication, regulate immune responses, regulate apoptosis, and so on. So if we put in this figure, and I focus just in the type I interferon, there is an intracellular pathway. Because of time, I will not go through it. It has been reviewed by many studies. The main point is here that when the signals induced by the interferon receptor will go to the nucleus, will activate these families of interferon-stimulated genes. So again, the question was if this is also existing in the trophoblast. So we have the induction of interferon beta. Is interferon beta binding to the receptor in the trophoblast? And what are the interferon-stimulated genes that are produced? So what we did is we stimulated the trophoblast cells with interferon beta, and we did a gene array to see if there is induction of interferon-stimulated genes. And the answer is yes. There is a high number of interferon-stimulated genes that are induced in the placenta and in the trophoblast. And they have multiple functions, including dendritic cell maturation, interferon signaling, and antiviral response. And this is just an example of the validation of those genes induced by interferon, including 6EL10 or IP10, an antiviral gene, IG20, and a pro-apoptotic gene, such as TRAIL. They can be specifically induced by interferon in the trophoblast. So if now we put the whole picture together, including the data that I showed you at the beginning, LPS binds to TLR4, induce TRIV, IRF3, IRF3 induce interferon beta is secreted, binds to its receptor in an autocrine and paracrine way, and interferon receptor activates the intracellular pathway, would induce the expression of these ISGs, and these ISGs will have a local or potentially systemic response. In addition, what we are finding is that interferon will induce the expression of intracellular modulators such as SOX1, and SOX1 will prevent the inflammatory pathway in the trophoblast and consequently enhancing the production of interferon beta. So the role of the placenta's interferon, based on these ISGs, include an antimicrobial, included an antiviral, and includes an immune modulatory signals. So the next aspect that we wanted again to discuss is what is the interferon, or what is the importance of the interferon in maintaining this homeostasis in response to microbial signals. And the experiment, one of the first experiments that we did in order to respond to this question, of course, is using the interferon receptor, the type I interferon receptor knockout mice. And what we did is we exposed wild-type mice to these doses of LPS that I mentioned before, and we look at the pregnancy outcome. And the same dose we inject to the mouse lacking the interferon receptor, the type I interferon receptor. And that is what we found. We injected on day 15, so we were looking if there is an effect on the preterm birth or pregnancy outcome. So if we have a wild type that can secrete and respond to interferon beta, when they are treated with LPS, we see minimal effect on those mice, on the pregnancy of those mice. But I want you to pay attention to this, the right side of the slide, where we have a mouse that can secrete interferon beta, but it doesn't have the receptor, therefore cannot respond to interferon beta. When we inject the same concentration of LPS as in the wild type, now the immune modulation is gone, and almost 80% of the pups will be delivered either early or they will die with the same concentration of LPS. These data suggest that whatever will affect the response by the type I interferon receptor will have a potential detrimental effect. In other words, if there is no inadequate response by to the interferon beta, pregnancy will be affected. These experiments were done with the interferon receptor knockout. So the question is, what in nature could affect the interferon receptor response, or the type I interferon response, and consequently lead to a pregnancy complication? And the answer, after searching for different factors, is viral infection. And how is that? So we did an experiment, again, taking a wild-type mice, where we inject MHV68, that is the mouse herpes virus, in day 8.5 of the pregnancy, so an early stage of the pregnancy. And we wait until day E15.5. That will be in humans early third trimester, late second trimester. And at that time, we inject the same concentration of bacteria that I described in the previous experiment. And we follow the pregnancy outcome. So here, I'm showing you with the black dots is the mice injected with the virus, and nothing happened to the pregnancy. When we inject the LPS, as I showed you before, there is a mild effect on the pregnancy. However, in the mice that were infected with the virus and then given the same dose of LPS, all of them were terminated early, or all of them were dead. So the virus was doing something to the immunological response in the placenta that was leading, or at the implantation site, that was leading to the termination of the pregnancy to the same level of stimuli. And when we look at the cytokine production and so on, when we give the virus alone and we look at IL-6, that is a factor that induces preterm birth and potentially fetal mortality, we see that there is no IL-6 in those mice. But if the mouse was exposed to MHV68 to the virus and we give the same doses of LPS, now you have a massive expression of IL-6. So what is doing the virus that is changing the response to LPS? And the answer is inhibiting interferon beta production. So we look in vitro at the cells infected with MHV68, and we look the levels of interferon production following LPS, and we see that it's suppressed in vitro. In vivo, in the placenta that was infected with MHV68 and was given LPS, we saw that there was IL-6 production, but there is an inhibition of interferon beta production. And when we look at the pathway that we know is critical for the induction of interferon, the TRIF, TBK1, and IR3, what we found is that when a virus infects the placenta, suppress the phosphorylation of IRF3, a critical step in the production of interferon beta. So the virus, by suppressing interferon beta, is now allowing an inflammatory response to the same bacterial product. And that's what we define as the two-hit hypothesis for preterm birth, or for the inflammation at the implantation site. In normal conditions, we discussed before, LPS or bacterial products will induce the expression of interferon beta, and that is the normal pregnancy. However, if there is a viral infection, the virus will suppress type I interferon pathway, and now the same TLR4 activation will induce a pro-inflammatory response responsible of the initiation of labor, or the preterm birth, or the fetal death. With this aspect, we further elucidated whether indeed the placenta is necessary for the protection of the fetus against viral infections. And again, this is the work done recently by Palomi, Jia Yuan, Karen, Xian Yong, and Yihao Ding. So the first question we wanted to know is the role of the placenta, or placental interferon during a viral infection in protection to the fetus. So what we did is we took wild-type mice. Again, we infected with MHV68, and we look at the viral titers in the placenta as well as in the fetus. So one of the first things that we found looking to viruses in the placenta is that the placenta gets infected with the virus. However, they have low titers. In other words, the placenta, although it's infected, is able to control the infection. When we look now in the fetus, we don't find any viral titers. In other words, although the placenta is infected, no virus are able to reach the fetus. The placenta is capable of controlling the virus and preventing the transfer or the movement of the virus towards the fetus. And again, we know that the placenta is susceptible to many viruses because we have tested all different types. They get infected, but it has a high capacity of control. And this is a figure comparing two cell types, trophoblasts in blue and stroma cells in green, looking infection with MHV68. As I mentioned before, both cell lines are infected, but while the placenta controls the viral titers and maintain low titers, stroma cells, they are infected, but they have not the capacity to control that viral infection. So next we wanted to know whether if there is no the interferon receptor signaling, in other words, if there is no the response to the production of interferon beta, whether in those cases the placenta will lose the capacity to protect the fetus and against a viral infection. So for this, we use again the interferon receptor knockout mice comparing to the wild type mice, and to the tube mice, we injected MHV68. First, we look at the placenta. And again, the placenta in the wild type mice is infected. However, if the embryo and the mother are knockouts, now the placenta has significant increase in viral titers. Again, this is logarithmic scale looking at the viral titers. So it's a huge amount of virus that are now present in the placenta. In other words, has lost the capacity to control viral replication in the absence of the interferon receptor signaling. What happened to the fetus? The wild type, there is no virus in the fetus. In the interferon receptor knockout, the fetus is now infected with the virus. It's really the placenta and the signals from the placenta responsible for this protection. So what we did is we cross a father that is an interferon receptor knockout and the mother that is a wild type, but there is an heterozygote. In other words, it can respond to the interferon receptor signaling. This cross will give us, in the same mother, in the same uterus, some fetus who are wild type, they can respond, and some fetus that are knockouts and therefore cannot respond. We injected with MHV68 and we look at viral titers in the placenta as well as in the fetus. Let's look at the placenta first. If the embryo and the mother are wild type, similar as we saw before, and again, this is in the same uterus, those wild type mice, the wild type fetus, the placenta is infected but low titers. In the same uterus, if the fetus is knockout, although the mother is wild type, but because the placenta or the fetus is knockout, they have lost the capacity to control the viral infection. And now there is high titers in the placenta. What happened to the fetus? Again, wild type mother, wild type placenta, no virus in the fetus. The mother is wild type, so it can respond, but the placenta cannot respond now while the fetus is infected. So when the placenta is incapable of response to the viral infection, although the mother is responding, the protection to the fetus is lost. So placental derived type I interferon protect the fetus from viral infections. So what is the mechanism mediating the placenta and antiviral infections? And again, is the ISGs, and we found those, we have characterized many of those who are associated with the antiviral response. And again, we know those are important because when we look in the interferon receptor knockout, again, that the fetus has been infected now, we see that it has a lot of viruses in the placenta, as I mentioned to you before, in the knockout, where there is no in the wild type. We look now at the interferon response. Both wild type and knockout are producing high levels of interferon beta. We see 126-fold change in the knockout. So it's able to recognize the virus and to produce a lot of interferon beta. However, the downstream of interferon is the problem. Because while the wild type is producing antiviral products, antiviral signals, such as RIG or ISG20, the knockout is not able to mount that response and therefore is not able to protect the fetus. So to put it in a graphic, we have the recognition, the induction of interferon beta. Interferon beta binds to the receptor and induces all these ISGs who prevent viral replication and protects the fetus. If that response goes, there is no protection and there is viral replication and fetal infection. And the last part of my talk is where we started the question at the beginning. Why pregnant women are at higher risk of mortality during viral infections? And we say it's not because they are immune suppressed. So if they are very strong, if they have a very strong immune system, why still they are more susceptible to mortality and to complications in the presence of viral infections? And again, Karen Rasico leads the study to answer this question. So how we found out? Again, so if we inject a virus to a wild-type mice, they will not be, the mother will not be affected. So they survive because both embryo and mother can secrete and respond to interferon beta. So there is no effect on survival, as indicated before. But if we do exactly the same, we inject MHV68, the same virus, to the knockout mice. In other words, they produce interferon, but they cannot respond to interferon. What we found is that there was mortality within 5 days after the injection of the virus. And here came the first surprise, and they're very interesting, because there was major differences between the non-pregnant and the pregnant knockout mice. What do I mean with that? If we inject the virus to a non-pregnant interferon receptor knockout mice, they will survive the virus for a long time. But if we inject the same virus to the same type of mice that were pregnant, they will die in 5 days. This data suggests that factors that may affect the interferon receptor signaling or the responses of the interferon receptor in the placenta during pregnancy will have a detrimental consequence to the maternal survival. What is the difference between the pregnant and non-pregnant? Of course, is the presence of the fetal placental unit. So our question was whether the increase in maternal mortality during pandemics is due to changes in the signals from the placenta or fetal unit. So in order to answer that question, we did what is called embryo transfers. So what we did? We took a mother that is interferon receptor knockout, and we transferred placentals from wild-type mice. In this case, the mother produced interferon, but cannot respond to interferon beta. The placenta produced interferon and can respond. And at the same time, we had, as before, the mother who is interferon receptor knockout, and we transfer a placenta where it's also interferon receptor knockout. We inject the virus to these two types of mice, and we monitor their survival. Let's look first at the model where we have an interferon, like I showed you before, an interferon receptor mother transfer an interferon receptor embryo. In the presence of the virus, the red line is showing you that the mothers will die between 5 to 6 days, as I showed you before. So now let's look what happened when we have the same mother that is interferon receptor knockout. But now it has embryos who are wild type. In other words, produce interferon and can respond to interferon. When we give them the same virus, now the mother will not die and is protected. So the response of the placenta to the virus is capable of preventing the maternal death. Or, if we want to put it in other words, the lack of the response, or the abnormal response of the placenta, is the one responsible of the mortality of the mother during that viral infection. And again, we see changes in the systemic organs of the mother, where if there is an interferon knockout mother, an interferon receptor knockout embryo, there is a major accumulation of viral titers in the spleen of the mother. Those numbers are significant decrease, almost to the normal levels, if the placenta is wild type. To summarize, placenta interferon beta protect the fetus and the mother for viral infections. The placental signals induced by interferon receptor has the capacity to control microbial infections, bacteria, and so on, modulates and regulates antiviral effectors, and an area that I didn't discuss and we're working, it produces a lot of immunomodulatory signals that are responsible for the differentiation and the function of immune cells at the implantation site. The placenta may be a preferred site for viral replications, but in general, the placenta has a high capacity to control the viruses through the induction of type I interferons. Neonatal complications, such as autism, microcephaly, and allergies, may be the result of abnormal responses during the fetal development. And I want to thank all the people who have contributed again to this work, and I hope this presentation will answer some of the questions associated why viral infections are detrimental for the pregnancy.